What if a single factor significantly increased your risk for most major diseases? Aging is identified as this primary risk factor. It contributes to cancer, heart disease, and neurodegeneration. This impacts health and longevity across populations. Our cells possess intricate repair mechanisms. However, these vital systems decline with age. This leads to a fundamental biological question. Why do our cells stop repairing themselves effectively? Understanding this process is crucial for future medicine. Cells constantly endure internal and external assaults. Metabolic processes within the cell generate harmful byproducts. Environmental stressors, such as UV radiation, also inflict damage. This damage primarily targets the cell's DNA. The body's intricate repair systems continuously work to fix these issues. However, DNA damage accumulates progressively over a lifetime. This constant onslaught eventually overwhelms the repair mechanisms. They can no longer effectively maintain genomic integrity. This leads to a significant decline in overall cellular function. Chromosomes contain protective caps called telomeres. These structures are found at each chromosome's end. Telomeres safeguard genetic information during cell division. With each division, these telomeres progressively shorten. This shortening occurs naturally over time. Eventually, telomeres reach a critical, short length. This signals the cell to stop dividing. The cell then enters a state called cellular senescence. Senescent cells remain metabolically active. However, they no longer replicate. This process contributes significantly to aging. Cellular senescence is a state where cells stop dividing. These cells remain metabolically active, however. Senescent cells contribute significantly to the aging process. They release a complex mix of harmful molecules. This is termed the senescence-associated secretory phenotype. These molecules include pro-inflammatory cytokines and proteases. Such secretions create a local inflammatory environment. This chronic low-grade inflammation, or inflammaging, damages neighboring healthy cells. It also disrupts normal tissue function. This persistent cellular stress accelerates tissue decline. Mitochondria are essential for cellular energy production. Their function declines as organisms age. This decline results in less efficient energy generation. Cells produce less adenosine triphosphate, or ATP. Aged mitochondria also generate more reactive oxygen species. These molecules cause oxidative stress within cells. Oxidative stress damages vital cellular components. This damage impairs the cell's ability to repair itself. Damaged mitochondria further reduce energy output. This creates a cycle of declining cellular health. Reduced energy and increased damage hinder overall cellular maintenance. Autophagy is a fundamental cellular recycling process. It systematically clears damaged organelles and misfolded proteins. This mechanism maintains overall cellular health. With advancing age, the efficiency of autophagy declines. Lysosomes, crucial for cellular degradation, show reduced activity. Consequently, damaged cellular components accumulate inside cells. This cellular waste impairs normal physiological functions. The buildup contributes directly to cellular dysfunction. It also accelerates the aging phenotype. An impaired system cannot remove cellular debris effectively. Stem cells are crucial for repairing damaged tissues. They replace old or injured cells throughout life. With advancing age, stem cell function declines. Their regenerative capacity significantly diminishes. The total number of active stem cells also decreases. These remaining cells often become less responsive. This leads to slower and incomplete tissue repair. Wound healing becomes less efficient in older bodies. For instance, muscle regeneration is severely compromised. The body's ability to maintain its integrity is reduced. Aging involves a persistent cellular challenge. This challenge is chronic, low-grade inflammation. Scientists term this phenomenon inflammaging. It is not an acute protective immune response. Instead, it is a sustained, subtle inflammatory state. This state contributes to widespread tissue damage. It also impairs normal tissue function. Inflammaging creates a destructive feedback loop. This loop exacerbates existing cellular and molecular damage. It prevents efficient repair mechanisms from operating. The systemic effect accelerates the overall aging process. It impacts nearly all organ systems in the body. Cells also experience epigenetic changes. These modifications alter gene expression. They do not change the underlying DNA sequence. Epigenetic markers accumulate throughout life. This accumulation disrupts normal cellular functions. Gene regulation becomes less precise with age. For instance, DNA methylation patterns can shift. Histone modifications also become altered. These changes affect which genes are active. They can silence essential repair mechanisms or activate genes linked to cellular decline. The precise causal links are still being mapped. This area remains a focus of scientific research. Research now targets specific hallmarks of aging. Scientists develop interventions to counteract these cellular processes. One promising approach involves senolytics. Senolytics are compounds designed to selectively remove senescent cells. These persistent cells contribute to chronic inflammation and tissue damage. Another strategy focuses on autophagy enhancers. Autophagy enhancers improve the cell's waste removal system. This restores efficient cellular maintenance and function. These targeted therapies aim to mitigate age-related decline. The goal is to extend healthy lifespan and improve vitality. Understanding these cellular mechanisms is vital. This knowledge opens doors for extending healthy lifespan. This concept is termed health span. The goal is to sustain good health for a longer duration. The focus shifts from treating specific diseases. Research now aims to prevent aging itself. This proactive approach targets the root causes of cellular decline. It seeks to maintain cellular repair capabilities. Such prevention could significantly improve quality of life. It also reduces the burden of chronic illness. We have identified many fundamental mechanisms of aging. These include telomere shortening and cellular senescence. Our understanding of these individual processes is robust. However, the complete interplay among these mechanisms is still being investigated. Optimal interventions for aging remain a critical research frontier. Scientists continue to explore how these complex cellular networks function together. The full scope of their interactions is not yet entirely understood. This represents a significant area of ongoing scientific inquiry. A central question persists in this field. Whether we can truly reprogram our cells for indefinite repair remains a major scientific endeavor. This represents a significant goal for future biological research. Thanks for watching. If you enjoy clear science explained with strong visuals and evidence, subscribe for more long-form science videos.